You know that transformation that happens when you take something as simple as flour, water, and yeast?
Hmm.
It starts as this sticky, unassuming blob, and then almost like magic, it starts to grow, to breathe, kind of inflates, it becomes dough. It's such a basic act, baking bread, but it feels ancient, almost mystical.
It definitely taps into something primal, that whole
Mhm.
transformation. But yeah, while it feels like magic, it's actually deeply rooted in some really fascinating science, a complex interplay of biology and chemistry, all working together to turn that simple mix into the airy, flavorful bread we love.
Absolutely. And that's exactly what we're diving into today. We got this fantastic stack of sources from a listener who's just incredibly curious about the science behind it all.
Ah, great.
So, our mission in this deep dive is basically to pull out the most important nuggets of knowledge and insight from all these sources, articles, research, notes, to give you a shortcut to being really well informed about the science of how dough becomes bread.
Okay, so we're going to unpack the core stuff,
Hmm.
starting with the real basics, the biology of yeast and fermentation. Then we'll get into how something like temperature acts as the sort of conductor directing everything.
Right.
We'll look at the surprising role of pH, how that affects the crust and flavor, how different ingredients change the outcome, the chemistry, the texture, and even how that affects flavor retention.
Oh, interesting.
And we'll definitely touch on the unique world of sourdough. That's a whole different ecosystem.
It really is a universe in every loaf. Okay, so let's get started. What's the absolute fundamental science? Why does dough get bigger? Why does it rise?
Okay, so at its core, in breads made with yeast, that visual change, you see the dough expanding, getting lighter, we call that leavening or just, rising. And it's pretty much entirely thanks to the activity of yeast.
Right, the yeast. One source described it as having a serious sweet tooth, which I thought was a fun way to put it, like tiny cookie monsters hunting for sugar.
Tacos. Yeah, that's actually not a bad analogy. Yeast, specifically baker's yeast, Saccharomyces cerevisiae, they're living, single-celled fungi. And like any living thing, they need food. Flour provides that food, but not directly as simple sugar. It's mostly starches.
Complex carbs, right.
Exactly. Long chains of simpler sugars like glucose, sucrose, maltose. Yeast have these enzymes, little biological tools, that break down those big starch chains into smaller sugars they can actually digest.
Okay, so they're eating the sugar that comes from the flour breaking down. What happens then? What are they like putting out that makes the dough rise?
Well, the magic happens through fermentation. Initially, when there's oxygen around, right after mixing, they respire, producing CO2 and water. But pretty quickly in a dense dough, that oxygen gets used up.
Uh-huh.
Then they switch to anaerobic fermentation. It's less efficient for them energy-wise, but it's key for baking. During fermentation, they convert those sugars mainly into two things: ethanol, which is alcohol, and critically for the rise, carbon dioxide gas.
CO2.
So they eat sugar and basically breathe out CO2 gas. Pretty much, yeah. That CO2 gas is the engine making it rise. It gets trapped inside this elastic network that's formed by the gluten proteins in the dough. When you need dough, you're developing that gluten.
Right, making it stretchy.
Exactly. Think of it like building millions of tiny, super stretchy balloons throughout the dough. As the yeast pumps out CO2, these little gas bubbles inflate those balloons,
Yeah.
and the whole dough mass expands.
So the rising is literally the dough inflating like a big web of tiny balloons.
Exactly. But it's an incredibly intricate network. And the strength and elasticity of that gluten network are just as vital as the gas production. If the network's weak, it can't hold the gas, the
Yeah.
bubbles just pop, and you end up with, well, flatbread.
Okay, that makes total sense. Now, the sources mentioned proofing and fermentation are sometimes used interchangeably. That can be a bit confusing. Serious Eats had something on this.
Yeah, they clarify it well. They point out that while proofing usually refers to that final rise, after you've shaped the loaf right before baking.
Uh-huh, the last step.
Right. But the underlying biology, the chemistry, it's the same process happening throughout all the rising stages.
Yeah.
Whether it's that first big rise or the final proof, the yeast is doing the same fundamental thing.
So, eating sugar, making CO2 and ethanol, the internal chemistry doesn't change, just the stage, we call it.
Exactly.
Hmm.
It's all fermentation. Biologically speaking, the terms often just signify where you are in the bread making process.
Got it. And speaking of stages, the sources really emphasize one phase, bulk fermentation.
Oh yeah, bulk fermentation is critical. That's the step where the entire batch of dough, after you've mixed and kneaded it, just sits and ferments as one big mass before you divide it up and shape it.
Okay, sounds simple, just letting it sit. But why is it so critical? The sources make it sound like this is where the real magic happens.
Well, it is. It's crucial for two main interconnected reasons that really set the stage for the final loaf. First off, flavor development. We said fermentation produces CO2, but also ethanol and a whole host of other aromatic compounds.
Right, the tasty bits.
Exactly. During bulk fermentation, especially once the oxygen is gone and the yeast shifts gears metabolically, the production of these flavor compounds really ramps up.
So letting the whole dough blob sit there for a while is basically building taste.
Precisely. If you rush the bulk ferment, yeah, you'll get gas, but you won't get much flavor complexity. A longer, properly managed bulk ferment gives time for those subtle, nuanced flavors, the alcohols, esters, organic acids to develop. That's what gives bread its characteristic depth of taste and aroma.
Fascinating. So it's not just about getting bigger, it's building the flavor profile. What's the second big reason?
Structure development.
Mm.
Kneading starts the gluten network, right?
Right.
But bulk fermentation lets it mature. The gluten strands relax, become more extensible, more stretchy, but also stronger in a way. The CO2 gently inflates this network, organizing it.
Ah, okay.
This helps the dough hold its shape better later, trap gas more effectively, and ultimately leads to a better texture, a better crumb in the final baked bread.
Building flavor and the internal architecture. Makes sense. Serious Eats had that example, the workhorse loaf. They said, what, 1.5 to 2.5 hours bulk ferment at room temp, aiming for 2 hours?
Yeah, that specific timing shows it's not just wait till it doubles. It's a controlled process with an optimal window for that balance of flavor and structure. And that phrase, comfortable room temperature, is key. Because temperature, that's probably the single biggest controller of this whole process. It's the conductor of the fermentation orchestra.
That seems intuitive, right? Yeast is alive, it cares about temperature. Atlas Scientific really emphasizes this.
They do. Atlas Scientific explains temperature has this direct, really profound impact on the rate and efficiency of yeast activity. All those enzymes inside the yeast doing the work. Their function is super temperature dependent. Generally, warmer temps mean faster reactions, faster sugar use, faster CO2 production.
But like you hinted, warmer isn't always better, is it?
Definitely not. Yeast has its happy place, its optimal temperature range. Go too hot, say above maybe 9500 Fahrenheit for many strains, and the yeast gets overworked. Its metabolism goes haywire.
Okay.
This can actually produce undesirable off flavors, sometimes kind of unpleasantly yeasty or overly alcoholic. And if you go really hot, like 140F or more, the heat literally cooks the yeast. Their proteins denature, they die.
And dead yeast means no rise.
No rise at all. Now, on the flip side, if it's too cold, the yeast gets lethargic, as Atlas Scientific puts it. Everything slows way, way down.
Right.
You get slow fermentation, minimal CO2, incomplete conversion of sugars. The dough just kind of sits there sluggish. Extreme cold can even make the yeast cells clump up and sort of fall out of suspension, making it harder for them to find the sugars.
Yeah.
So you need that Goldilocks zone, not too hot, not too cold, for the yeast to work efficiently, leading to optimal product formation. But that brings up the question Serious Eats asked. Why not just make the dough really warm so it proves super fast, get bread quicker? Tacos. It's tempting, isn't it? Especially when you just want that bread. But rushing bulk fermentation with high heat has some serious downsides for the final quality.
We talked about flavor, less flavor if it's too fast.
Exactly. Those really nice complex flavors, the subtle alcohols and esters. They're produced most significantly after the oxygen's gone and the yeast is deep into anaerobic fermentation. If the dough gets too warm and ferments too fast, the yeast just prioritizes pumping out CO2 for rapid growth. It doesn't spend as much metabolic energy making those nuanced flavor compounds, so you get lots of lift, fast, but a shallower, less complex flavor. Speed kills the taste, basically.
More bubbles, less deliciousness. What about the structure? Does heat mess with the gluten?
It does, but sort of indirectly. Gluten development, that stretching and strengthening, is less sensitive to temperature changes than yeast activity is. So if the yeast is going crazy fast because it's too warm, it's pumping out CO2 gas much faster than the gluten structure can comfortably relax, stretch, and organize itself around those expanding bubbles.
Like blowing up a balloon way too quickly.
Perfect analogy. The little CO2 bubbles overinflate and pop because the gluten around them isn't ready to stretch that fast or that far. This actually weakens the overall gluten network. Dough that's fermented too hot and fast can feel sticky, weak, hard to handle when you try to shape it. And then in the oven, because the structure is compromised, you don't get as good an oven spring, that final puff. The loaf often ends up flatter, maybe denser. Serious Eats called it less sexy-looking bread, and yeah, they're right.
Laughs. Okay, noted. Avoid unsexy bread.
So finding that optimal temperature is really about balancing everything, the yeast speed, the gluten development, and the flavor creation all happening at the right pace.
Exactly. It's not just about maximizing CO2, it's about synchronizing multiple processes. And the sources give some temperature examples. Atlas Scientific mentions general bread fermentation might be fastest for CO2 between 85-90 F.
That sounds pretty warm, right, but Serious Eats recommended more like 75F for their workhorse loaf, taking longer, 1.5 to 2.5 hours.
Yeah.
That's quite a bit cooler. It is. And that difference is deliberate. That 85-90F might give the fastest rise, but like we just said, speed can hurt flavor and structure. A slightly cooler temp, like 75F, slows the yeast down just enough to allow for better flavor development and give the gluten network more time to strengthen properly as it inflates. Many artisan bread recipes actually favor even cooler bulk ferments, or use cold fermentation like in the fridge for the final proof to really push flavor development.
Ah, okay. That's why people put dough in the fridge overnight. Slow it way down for flavor.
Exactly. Cold fermentation, say around 40-45 F in the fridge, dramatically slows yeast activity, minimal CO2. But it gives enzymes loads of time to break down starches, and it lets those slower flavor-producing metabolic pathways really shine.
That connects to other things too, like brewing. Atlas Scientific mentioned lager beers use cold fermentation, 48-58F, for a clean, crisp taste, minimizing those fruity yeast notes.
Right, while ales ferment warmer, 68-72F faster, producing more complex flavors from yeast byproducts. Same principle. Temperature controls the speed and the type of metabolism, shaping the final flavor.
Managing this at home can be tricky, though. Serious Eats had some tips.
Yeah, simple things. If your kitchen's hot, maybe put the dough in a cooler spot, like a basement or low cabinet. If it's cold, maybe near a pilot light. Or use a proofing mat for gentle warmth. Even just setting your house thermostat to around 75F can help.
And Atlas Scientific added that fermentation itself creates heat. It's exothermic.
That's a really important point, especially for larger batches. The dough mass can actually warm itself up as it ferments. Pros have to manage this with controlled temperature rooms or special containers.
Can you, change the temperature partway through? Start cool, then warm it up?
Atlas Scientific says yes, that's sometimes done. Maybe start cooler for flavor, then warm it slightly to boost the rise later. Takes a bit of know-how, understanding your dough and environment.
Okay. So temperature is the maestro for fermentation, conducting the rise and building initial flavor. But then, when that dough hits the hot oven, a whole different set of reactions kick in, especially the Maillard reaction. And our sources say pH plays a huge role here.
Yeah, absolutely. The Maillard reaction is fundamental to what happens during baking. The Reddit source defines it as this complex series of non-enzymatic reactions, meaning yeast isn't directly involved, that happen at high temperatures between amino acids from a flour's protein and certain types of sugars, called reducing sugars.
And this is what gives bread that beautiful brown crust, right? But it's more than just color.
Oh, much more. The Reddit post mentions it creates hundreds of different molecules. These are responsible for a huge part of the aroma and flavor of the crust, those toasty, nutty, savory, sometimes slightly sweet notes. It's complex flavor creation happening right there on the surface.
And the pH, the acidity or alkalinity of the dough surface, affects how well this reaction happens.
Profoundly. This is where it gets really cool, chemically speaking. The Reddit post explains the Maillard reaction happens more readily in alkaline, meaning high pH environments.
Why? What's the chemistry behind that?
It comes down to the amino acids. In acidic, low pH conditions, the amino groups on those acids tend to hold on to an extra hydrogen ion they're protonated. This makes them less reactive. But in an alkaline, high pH environment, they're more likely to lose that hydrogen ion they get deprotonated.
Okay, so losing a hydrogen makes them more reactive. How?
Yeah, losing that proton leaves the nitrogen atom in the amino group more electron-rich. This boosts its nucleophilicity. Think of that as how strongly it's attracted to positive charges on other molecules. Sugars have positively charged spots, so deprotonating the amino acid makes it much more eager to chemically attack and bond with the sugars when heat is applied, kicking off that whole Maillard reaction cascade.
Wow. So basically, high pH primes the amino acids, gets them ready to react with sugars in the oven's heat, leading to faster, more intense browning and flavor. The Reddit post had that amazing picture comparison with pretzel bread.
Yes, that experiment shows it perfectly. Same dough, but different surface treatments right before baking. One piece dipped in a strongly alkaline 4% sodium hydroxide solution that's food-grade lye. Another in a weakly alkaline 3.5% sodium bicarbonate solution, just baking soda. And a control dipped in plain tap water, which is near neutral pH.
And the difference in the photos is just stark, isn't it? The lye one has that super dark mahogany brown pretzel crust. The baking soda one is nicely brown, way darker than the control. And the water one is really pale.
It's a crystal clear demonstration. Upping the surface pH before baking dramatically speeds up the Maillard reaction, giving you much richer color.
The comments on that Reddit post got into the different alkaline things, lye and AOH, strongly alkaline.
Oh yeah, it's potent stuff, sodium hydroxide, very caustic. The comments rightly stress. If you use food-grade lye, safety is paramount. Gloves, eye protection are musts. And when dissolving it, you add the lye to cold water, slowly. Never water to lye, never hot water, can react violently. Good ventilation is key too. Rinse skin immediately if it touches you. Pros often use cold lye solutions for better control and safety.
Yikes. Okay, handle with extreme care. Baking soda, NaHCO3, is the weakly alkaline option. Boiling dough in baking soda water is the common home pretzel trick, right?
Exactly. It's much safer than lye. And boiling it raises the surface pH enough to get decent browning, much better than plain water. It's the go-to for most home bakers making pretzels.
And then there was that middle ground mentioned in the comments. Baked baking soda.
Yeah, that's a neat trick. You take regular baking soda, sodium bicarbonate, and bake it in a low oven, maybe 250 F, for an hour or two. Heat converts it to sodium carbonate, Na2CO3. This stuff is much more alkaline than plain baking soda, but not nearly as caustic as straight lye.
So it boosts the pH more than regular baking soda, gets you closer to the lye effect, but it's way safer to handle.
Exactly. It's a great compromise. You get a darker, richer crust than with just baking soda, often good enough for that pretzel look and taste without needing the full hazmat suit for lye.
Fascinating. Now, besides the color, the Reddit post said this alkaline treatment also changes the crust texture and even the inside.
It does. Dipping the dough in water starts a partial gelation of surface starch. The starch on the outside absorbs water, swells up, gets kind of gelatinous. This actually helps the Maillard reaction too, by making sugars more available. But it also changes the structure.
How so?
It causes the crust to set much more quickly once it hits the hot oven. That gelled layer firms up fast. And if the crust sets early, it limits how much the dough can expand in that initial burst of heat, what bakers call oven spring.
Ah, so by setting the crust faster, you're essentially putting a lid on how much it can puff up.
Pretty much. Less oven spring usually means a denser, chewier crumb, which is exactly what you expect in a pretzel or a bagel treated this way. You see it in the scoring marks in the photos too. The alkaline breads had these beautiful and wide cuts where the crust split cleanly. The plain water one, with less crust setting, had smaller and undeveloped cuts. It shows how the treatment dictates how the crust behaves and where the dough expands.
That's incredible. It's not just changing the surface color. You're fundamentally altering how the crust forms and the resulting texture inside.
Yeah, it's a powerful example of how a simple surface chemistry tweak impacts the whole baking process and the final bread.
And you mentioned sourdough earlier. Sourdough is acidic, low pH. Does that mean it hinders the Maillard reaction, makes it harder to brown?
Well, the Reddit comments speculated it could slow it down a bit theoretically because the low pH keeps amino groups protonated, less reactive. But sourdough crusts still get beautifully brown, right? That's partly because another browning reaction, caramelization, which is just the browning of sugars themselves at high heat, happens regardless of pH and contributes a lot to sourdough color. Plus, the long fermentation creates lots of sugars and amino acids anyway, plenty of fuel for both reactions.
Okay, so caramelization helps make up for any potential Maillard slowdown in sourdough.
Got it. Okay, we've covered yeast, temperature, pH, baking. Let's shift to the ingredients themselves. Beyond just feeding the yeast, how does the type of flour or starch impact the final bread, especially its smell, its aroma? Yeah, that's what the PMC study looked at. Specifically with gluten-free flours, things like corn, rice, oat, but also pseudo-cereals like teff, buckwheat, amaranth, quinoa, and different starches. They wanted to see how these affected the volatile compounds. Those are the molecules that create smell and flavor, both in the raw dough and in the baked crumb.
Like the chemical fingerprint of the bread's aroma, what kinds of compounds did they find?
They grouped them into two main types. First, fermentation compounds, mainly ethanol, the alcohol, and these things called 2 and 3-methylbutanol, direct byproducts of the yeast working.
Right, we know yeast makes ethanol.
Exactly. The second group was lipid oxidation compounds like hexanol, 1-pentanol, 2-monophore-decadenol. These come from fats in the flour reacting with oxygen, often helped along by enzymes or heat.
Okay. And did the ingredient choice change the levels of these?
Big time. In the raw doughs, the fermentation smells, like ethanol, were higher in doughs made from cereal flours, corn, rice, oat. Interestingly, the lipid oxidation smells were higher in the doughs made just from starches. But then, after baking and the crumb, things shifted. Fermentation compounds were higher in crumbs from those pseudo-cereals quinoa, amaranth, and the lipid oxidation compounds, much higher in crumbs from cereal flours, especially corn.
Whoa, so the ingredient affects not just what smells are made, but maybe which ones survive baking.
That's exactly it. The study notes a considerable reduction in those fermentation volatiles, like ethanol, from dough to crumb, because they evaporate during baking. They're volatile.
Makes sense. Alcohol boils off.
And this is where it gets really mind-bending. The amount that evaporates isn't just about boiling points. It's also heavily influenced by the resistance to mass transfer from the matrix to the air.
Okay, resistance to mass transfer. Break that down for me.
Think of it like this. How hard is it for those smell molecules to physically get out of the bread structure and into the air so you can smell them? The matrix is the bread itself, its internal structure.
So the bread's physical structure acts like a barrier, controlling how much aroma escapes.
Precisely. And the study shows this resistance depends heavily on the bread's macro and microstructure and texture. Different flours create fundamentally different structures. For instance, those pseudo-cereal flours, quinoa, amaranth, teff, buckwheat, they often make much more compact breads compared to breads made from starches or even regular wheat flour.
Why are they more compact? Denser.
Yeah, the study cites other research showing that bran particles, which you find in wholemeal pseudo-cereals, can actually puncture and break the gas bubbles that the yeast creates. This messes with the ability to trap gas efficiently, so you get less volume, a denser crumb.
Fewer intact bubbles means a less airy structure.
Right, and here's the kicker. That more compact, denser structure provides higher resistance to mass transfer. It makes it harder for volatile compounds to escape. So even though they're denser, the breads made with pseudo-cereals like amaranth actually retain more of those yeast-produced fermentation compounds, like ethanol, in the final crumb.
Kick out! That's completely backwards to what I'd think. You'd assume airy bread traps more flavor, but the dense structure actually holds on to the volatile stuff better.
It's an amazing finding, right? Shows that complex link between physical structure and flavor chemistry. The study's data was clear. Amaranth bread had the lowest ethanol evaporation, around 38%, and the smallest volume. Wheat starch bread was super airy, huge volume, but lost almost 87% of its ethanol during baking. The structure directly controlled flavor retention.
Wow, okay. What about the lipid oxidation compounds being higher in the corn crumbs? Is that structure too, or something else?
That seems to be more about the inherent chemistry of those flours. Cereal flours, especially corn, just tend to have more of certain fats and also higher activity of enzymes like lipoxygenase. This enzyme speeds up fat oxidation, producing things like hexanol. Pseudo-cereals like quinoa, on the other hand, have lower lipoxygenase activity and also contain natural antioxidants that slow down that process.
So the ingredient choice impacts flavor production through yeast and through other chemical pathways like fat oxidation, plus it impacts flavor retention through the physical structure it builds, layers upon layers.
Exactly. The raw ingredients aren't just passive building blocks. They're actively shaping the chemical reactions and the final sensory experience in multiple ways.
And speaking of complex experiences, we absolutely have to talk about sourdough. That feels like a whole different ballgame in terms of bread transformation.
It really is. Sourdough baking taps into much older techniques and relies on a fundamentally different biological engine compared to using commercial yeast.
The history is so cool. Our sources mention the San Francisco sourdough tradition, supposedly brought over during the gold rush. It's so unique that the main bacteria found there got named Fructolactobacillus sanfranciscensis after the city.
That's right. And the tradition even traveled north to the Klondike Gold Rush, leading to prospectors being nicknamed sourdoughs because they carried their starters with them, keeping them alive. They had to protect them from heat, though apparently freezing doesn't kill the culture.
Such a rich history, and you see it today with this amazing online community, people sharing starters, troubleshooting, reviving old strains, passing down starters for generations like Carl Griffiths.
It really speaks to the fascination people have with it. And yeah, the pandemic definitely boosted interest when commercial yeast got scarce. People rediscovered this traditional method.
So scientifically, what makes sourdough different from just using a packet of yeast?
The biggest difference is it's not just one type of yeast. Sourdough relies on a symbiotic community, a whole mini ecosystem of living organisms. It's mainly lactic acid bacteria, LM, and various types of yeasts, all living together and helping each other out in the starter.
So it's a team effort between bacteria and yeast.
Exactly. The Wikipedia source mentions key players. Besides S. sanfrancesensis, you find other LA, like Alpontis levolactobacillus brevis. These bacteria are the ones fermenting sugars to produce lactic acid and acetic acid. That's what gives sourdough its characteristic tangy, sour flavor.
Ah, hence the sour in sourdough.
Precisely. Then you have the yeasts. Common ones include Kazakhstania exigua, sometimes even strains of Saccharomyces cerevisiae, baker's yeast, and often Kazakhstani humulus. K. humulus is interesting because it can't eat maltose, a made sugar and flour, but it thrives in the acidic, high acetate environment the bacteria create.
Okay, so how do they work together? What's the symbiosis?
It's like a little metabolic partnership. The main bacterium, F. sanfrancesensis, is great at fermenting maltose, but not so good with other sugars like glucose. So acid-tolerant yeasts like K. exigua or K. humulus, which can't use maltose, step in and ferment the glucose released as starches break down. The yeasts produce the CO2 for leavening and ethanol for flavor. And in return, the yeast can provide things like amino acids or vitamins, growth stimulants that the bacteria need.
Wow, so they're basically feeding each other and creating this unique environment where they both do well.
Exactly. They establish this stable, acidic environment that keeps out undesirable microbes, but is perfect for their specific partnership. Traditional sourdough, used as the only leavener, type I, is usually a firmish dough, fermented maybe 6886F, and gets quite acidic down to pH 3.8 to 4.5.
Does the mix of microbes change over time when you start a starter?
Yeah, if you start one from scratch with just flour and water, it evolves. Initially, you might get all sorts of bacteria, but as the LAB produce acid, the environment gets selective, favoring the sourdough specialists like F. sanfrancesensis. The yeast tend to grow slower and reach lower populations, maybe 1/10% of the bacteria numbers, but they find their niche and the whole community becomes stable and well adapted.
And this whole complex community must be why sourdough tastes so different, right?
Absolutely. Sourdough yeasts are often less aggressive, more diverse than commercial strains. Working alongside the bacteria, they create a much wider range of flavor compounds. The bacteria contribute massively with the acids and other aromatics. Commercial yeast fermentation is pretty straightforward by comparison. Plus, the longer fermentation times typical for sourdough allow these complex flavors to fully develop.
That longer time means it's usually not great for a standard bread machine, I guess.
Generally, yeah, the cycles are too fast for a true sourdough, leavened only by the starter. People find workarounds, like maybe adding some starter for flavor alongside commercial yeast or just using the machine for mixing. But real sourdough usually needs that patient, longer fermentation. And getting that classic crust blistered, crackly, with those nice cuts, that often needs specific oven techniques, like using a Dutch oven or creating steam to control how the crust sets and allow for good oven spring.
It's cool how the sources point out similar fermented dough techniques all over the world. Danish rug brood, Mexican burrito, German pumpernickel, Ethiopian injera, Indian idlis.
Yeah, it really shows that using microbial fermentation is this fundamental human approach to transforming grains and creating unique foods everywhere. And the source briefly touches on potential nutritional aspects being researched, like sourdough possibly reducing certain hard-to-digest carbs, FODMAVs, or breaking down certain proteins found in wheat.
So maybe easier to digest for some people, another interesting layer.
Definitely. It's not just about history and flavor. The nutritional side is being explored too.
Okay, let's try and pull this all together. We've gone pretty deep into the hidden world of dough.
We really have. We started with the basics, the yeast and fermentation, driving the rise and initial flavor.
Then we saw how temperature is like the master controller, setting the pace, and that critical balance between rising fast versus getting good flavor and structure.
Then we jumped into the oven, looking at how surface pH dramatically changes the Maillard reaction, affecting color, crust flavor, how the crust sets, which impacts oven spring and the final texture inside.
Right. And we looked at the ingredients, how the type of flour or starch influences not just the fermentation flavors, but other pathways like lipid oxidation, and incredibly, how the physical structure they create determines how much of those aromas actually stay in the bread.
And finally, we dipped into sourdough, that ancient, complex ecosystem of bacteria and yeast working together, creating unique flavors over time that you just don't get with commercial yeast alone.
What's super clear is that none of this happens in a vacuum. It's all connected, isn't it? The kitchen temp affects the yeast, which makes flavor, but whether you taste that flavor depends on the flour's structure. The dough's pH affects the crust, which affects the crumb texture. It's all interacting.
It really is a complex biological and chemical symphony playing out in that simple mix of flour and water. Every single variable temperature, flour type, fermentation time, surface pH, it all feeds into the final loaf you hold and taste.
It's amazing to think about all that invisible activity. So, the next time you bite into a slice of bread, whether it's fluffy white sandwich bread or a tangy sourdough with that dark, chewy crust, maybe think about the journey it took. From those simple ingredients to this complex structure holding an orchestra of flavors shaped by tiny microbes, temperature, pH, and the very physics of the dough itself, what might you notice differently, knowing the science that got it there?
Yeah, and understanding these principles, it doesn't just make you appreciate bread more, it can really empower you in the kitchen, right? Trying different flours, playing with fermentation temps, maybe even starting your own sourdough, it becomes less about just following steps and more like conducting your own delicious experiments. The sources we looked at today are a great starting point if you want to dive even deeper into this amazing science.